genetic technologies. All right. So, what are molecular genetic technologies? There are advanced techniques used to analyze, manipulate, and understand the genetic material, the DNA and RNA of organisms. All right. So, let's look at some of them. The first one is gene transfer technology, or what we call transgenesis. What is it about? It involves incorporation of genes from one species into the genome of another species so that it functions stably in the recipient species and is transmitted from one generation to the next. So the animal that receives the gene of the foreign DNA is called transgenic animal. All right. So the methods involved, there are two methods that are involved in the process of transgenesis. The first one is a direct microinjection of foreign DNA to the pronuclei of fertilized eggs at the early stage of embryonic development. All right. So the next method is the infection of pre-implantation embryos with retroviral vectors. So these are two methods that are involved in transgenesis. All right. So we're going to look at the second technique, which we call DNA fingerprinting. DNA fingerprinting. So what is DNA fingerprinting? DNA fingerprinting, also known as DNA profiling or typing, involves graphically characterizing an individual's DNA obtained from any tissue. So you can just cut off tissue from an organism and use it to profile the DNA of that organism. So it's widely used in parentage insemination and also in forensic sciences. In animal breeding, DNA fingerprinting is mostly used in parentage identification in pedigreed populations. Right? So that is DNA fingerprint. So the next one we'll look at is marker-assisted selection. What is marker-assisted selection all about? That's mass. What is it about? So marker-assisted selection is a technique that uses molecular markers to select individuals with desirable traits, right, for the purpose of breeding. Right. So we have examples of molecular markers. Right. So there are several of them. So molecular markers are sequences of DNA in the genome that are associated with specific genes or regions of the genome linked to the traits of interest. Examples include microsatellites, or what we call simple sequence repeats, right, SSRs, and we also have another one we call single nucleotide polymorphisms, which we shorten as SNPs or SNPs. We also have another one that we call restriction fragment length polymorphism, that is RFLP or reflex. So these are common examples of molecular markers that are used in marker-assisted selection. All right. So the next thing we are going to be looking at is an overview of advancements in molecular biology, which has contributed a lot to the development of genetic engineering. So what are those advancements? The first one we're going to be looking at is the establishment of the structure and functions of the genetic material, which is the DNA, yes, which we know. Right. So the next one is development of techniques for separation and detection of DNA fragments. The example is electrophoresis. All right. So another one is a development of methods for multiplying sequences of DNA or genes. A typical example of that one is a polymerase chain reaction. All right. So we also have a development of DNA sequencing technologies. All right. There are different technologies for sequencing DNA. All right. So we also have discovery of enzymes which are capable of cutting up sequences of DNA at sequence-specific sites or sequence recognition sites. All right. So we also have the discovery of vectors which can carry foreign DNA fragment or gene into the host cells. So these are some of the advancements in molecular biology that has actually helped to shape genetic engineering, or what we call recombinant DNA technology. All right. So we're going to be looking at the reproductive technologies that we use in animal breeding. And of course, you know that the mechanism by which genes are transmitted from parents to offspring is through the process of reproduction. So we have two important technologies that we use. All right, artificial insemination is the first one, and another one is embryo transfer. There are also other ones that we also use, which includes semen sexing and the embryo sexing. We also have what we call embryo cloning. All right, so these technologies are very useful in the field of animal breeding and animal breeding and genetics. Sorry. So let's look at each of them one after the other. So the first one we are going to look at now is artificial insemination. So what is it about? It is simply the process of collecting semen from a superior performing male and then introducing that semen into a recipient female. That is done through the genital tract of the female, and that female must be in heat for that to work well. All right. So what are the steps involved in artificial insemination? What are the steps that we take? All right. So let's look at the steps involved in artificial insemination. So the very first step that we take in artificial insemination is first of all collect semen from a superior performing male from a population. All right. So after collecting semen, the next thing we do is to evaluate the semen for quality, and of course the semen must be of high quality. So after evaluating the quality, the next thing you do is you dilute the semen in order to increase its volume. All right. So after doing that, you can now deliver the semen to the receptive female. Right. All right. That is the female that is in heat. We're talking about the benefits of AI in breeding. One, it brings about rapid dissemination of genetic improvement, through the use of a superior performing sire, that is sire of high genetic merit. Another benefit is it increases the rate of genetic get. So from animal breeding perspectives, these are some of the benefits of using artificial insemination. All right. So let's look at another technology that we call embryo transfer technology. So what is embryo transfer technology? What is it about? It simply involves the collection of embryos from donor females and their transfer to recipient females. All right. So how is that done? How is that done? How is it done? So we have different methods of embryo production. Of course, before you do the transfer, you have to first of all produce a fertilized egg or fertilized ovum. So we have two processes that we use. Is either it is done inside the animal, that is the in vivo fertilization, or it is done outside the animal, which we call in vitro fertilization. That is the one that you carry out in the laboratory. So what is in vitro fertilization all about? So it involves so the first thing you do is a super ovulation of the donor through hormone administration. Of course, you have to inject hormone into the animal to let the animal get to ovulate. So after doing that, you now carry out artificial insemination of the donor female. Once that is done, you now flush the embryos in order to collect them, and then subsequently implantation of the embryos to recipient females immediately after recovering those embryos. Or you can also do that when you preserve the eggs, you can use them later for implantation into recipient females. So we have the second method that we call in vitro fertilization. So what is it about? That is the one that we carry out in the laboratory. How is it done? It involves a simple collection of eggs from ovaries, their maturation, and then fertilization in the laboratory. All right? Are you following? So the resulting embryos are then cultured. You have to culture them to a certain stage before you can now transfer them to the recipient females, right? Which you must have conditioned, and that should be done immediately, or can the fertilized egg can be frozen for later use, right? So what are the benefits of embryo transfer? What are the benefits in animal breeding? So one of the benefits is that it increases genetic gain, also increases the production of several progenies, from a superior performing dam. All right. So another importance is genetic conservation of endangered species. So the next technology is semen and embryo sexing. What is it about? So these two technologies simply involves the separation of the sex cells, that is the gametes, the sperm cell, which can contain either X chromosome or the Y chromosome. So you have to sort the male gametes based on whether each one is carrying X chromosome or is carrying Y chromosome. So one of the methods of doing it is by using what we call the flow cytometry. So how is that done? What is involved? The technique involves staining the sperm cells with fluorescence dye that binds to the DNA, and of course you know that the X chromosome is larger than the Y chromosome. So once they are stained, and then when you pass them through a laser beam, the X chromosome is going to emit a stronger fluorescent signal than the Y chromosome, because it is bigger than it contains more genetic material than the Y chromosome. So let's look at embryo sexing as one of the reproductive technologies. So what is it about? It simply involves the determination of the sex of an embryo before it is implanted into a recipient female animal. All right. So that is what it is. So how is it done? How can we do that? We can do that using a process we call karyotyping, which is geared towards confirming the presence of the sex chromosome, that is X or Y chromosome. All right. So the next one is embryo cloning, which involves the production of genetically identical copies of an embryo from a single embryo, and its major use in animal breeding is for conservation of endangered species. So what does the process involve? It involves simply the manipulation of an embryo at a very early stage of development, to produce multiple copies of the embryo that can develop into genetically identical organisms. So that is what embryo cloning is all about. All right. So once an embryo is cloned and you have multiple copies of it, you can now do what? Can transfer them into recipient females, where they can now develop into full organisms. So that brings us to the end of the reproductive technologies that we use in animal breeding for the purpose of genetic development of our farm animals.